I would like to introduce our today's speaker, Dr. Matt Kaeberlein. I met Dr. Kaeberlein at molecular biology aging courses in marine biology laboratories in 2001. Dr. Kaeberlein was almost completing his doctoral studies with Dr. Leonard Guarente at MIT, and he was serving as course instructor during the molecular biology of aging courses. And since then, he did several courses in the marine biology laboratory. After a conventional postdoctoral training at the University of Washington, he secured assistant professor position at University of Washington and rose to full professor rank. Currently, he's a professor in the Department of Laboratory Medicine and Pathology at University of Washington. Dr. Kaeberlein spent more than 20 years to study the basic biology of aging using biochemical genetics and molecular approaches. During this time, he published more than 200 papers with the high index 67 and I-10 index 165. These are a little bit outdated as of January 11, so I'm sure that the numbers are different. Okay, look at this number. His work received a total of 26,261 citations. If the Google Scholars, that says correctly, I think they definitely says correctly. And then his most cited 2000 Nature article achieved a 2,401 citation. Very impressive. Secured several federal and private grants, served multiple leadership roles at the University of Washington and within scientific societies, including the American Association for Advancement of Science. He is a fellow of the AAAS, the American Aging Association Fellow, past president, board of directors. In Gerontology Society of America, he was a fellow, Biological Science Chair Council, and the Federation of American Societies for Experimental Biology. He served as a board of directors. Dr. Kaeberlein currently serves as the director of the University of Washington Healthy Aging and Longevity Research Institute, director of Biological Mechanisms of Healthy Aging Training Program, director of University of Washington Nathan Schuck Center of Excellence in the Basic Biology of Aging, and founder and co-director of the Dog Aging Project, something that I am interested in. And I'm very happy that Dr. Kaeberlein will spend some quality of time with us. Without further ado, please join me for welcoming Dr. Matt Kaeberlein. Dr. Kaeberlein, floor is yours.
All right. Thank you very much for that kind introduction. I will share my screen. Hopefully, after all of this time, I've learned how to use Zoom. So hopefully you can all see the presentation. And so what I want to start today by talking about is give you sort of an introduction to the biology of aging, why I think that it's an interesting and important area of research. And then I'll focus a little bit more on a specific pathway that we're interested, the mechanistic target of rapamycin pathway, and a drug that is an inhibitor of that pathway. Tell you a little bit about work that we are doing and have done in laboratory mice. And then I'll finish up by telling you a little bit about the dog aging project and how we are trying to understand the mechanisms of biological aging in companion dogs. So I want to start again, sort of big picture, and talk a little bit about the way that sort of traditionally from clinical perspective and a biomedical research perspective, we have approached health and longevity. And really, the traditional approach has been focused on disease. And what I mean by that is that if you go to the doctor, usually they will, if you have a symptom, they will try to diagnose what that symptom means, what the disease is that is causing those symptoms. And then once you have a diagnosis, the focus becomes on either curing the disease or at least alleviating the symptoms. And that has largely been the way that we have approached health in the clinical side. And from a research side, if you look at the way that most of the federal funding for research is allocated in the United States and in other countries, the vast majority flows towards individual diseases. So within NIH, there is a National Cancer Institute, an Institute for Kidney Disease and Cardiovascular Disease, and even the National Institute on Aging, the NIA, where most of my research comes from. More than half of the NIA budget is focused on Alzheimer's disease. So it's really a disease-focused approach to health. And I think what often gets lost when we think about health from a disease first perspective is that all of these major causes of death and disability that are really the leading causes of death and disability in developed countries have at their root a single greatest risk factor. And it's not whether you smoke or how much you eat or how much alcohol you consume or whether you're overweight, it's how old you are. So age is by far, by at least an order of magnitude, and for some diseases, multiple orders of magnitude, the greatest risk factor for developing these diseases. And so if we can understand, what is it about biological aging that is at least permissive for these diseases? I think we can have a debate about whether biological aging causes individual diseases, but there's no question that it creates a physiological state that is permissive for these diseases. And if we can understand, what are the mechanisms of biological aging, we have the potential to actually target those mechanisms to hopefully delay the onset and progression of all of these diseases of aging at the same time, and the functional declines that go along with those diseases. And so this is what I think of as 21st century medicine. And I say that to differentiate it from what I think of as 20th century or even 19th century medicine, which is to focus on individual disease. It is my hope and expectation that within this century, we really will, from a research perspective and from a clinical perspective, start to think more about what is it about biological aging that is driving all of these diseases, and approach health from that perspective, which is really much more of a preventative approach than a reactive approach, than waiting until people are sick. Okay, so you can start to ask the question then, what would that mean if we were successful in this endeavor of actually targeting aging? What would that mean? And one way to look at this is just to look at life expectancy or lifespan. And so this is work that was originally done by J. Olshansky and colleagues to ask the question, what would it mean if we could cure individual diseases in a typical 50-year-old woman in the United States? And these numbers are a little bit out of date, but they're not going to have changed much. And so the CDC is actually quite good at keeping track of what people die from. And so it's fairly straightforward to ask, for a typical 50-year-old woman, what would the impact be on life expectancy if we removed all deaths due to cancer or all deaths due to heart disease? So that's what's shown here. And what you can see is that the effect on population life expectancy is actually quite small. It's much smaller than I thought it would be, before I had seen this data. If we could cure all forms of cancer today, right now, typical 50-year-old woman would get about 3 extra years of life expectancy. Same thing's true for heart disease. And even if we could cure both, that only leads to about 7 extra years of life expectancy. And I think, when you think about why that's the case, it immediately becomes obvious, right? If you only fix one disease, your risk of developing all the other diseases of aging continues to increase exponentially. And so the incremental impact of curing one disease is relatively small. And I want to just briefly take an aside here and make the point that, this is a population average that we're discussing here. At the individual level, curing cancer or curing heart disease is extremely important, right? If someone I loved had cancer, I would want that disease cured. So I'm not at all trying to minimize the impact of curing individual disease after they've already developed, but at the population level, the impact is actually quite small. And if you think about the amount of resources we have put towards the war on cancer over the past 3-4 decades, when you consider the impact at a population level, if we were successful, which we haven't been, at curing cancer, it's relatively small. Okay, so then we can ask the question, what would the impact be if we took this 21st century approach, right, of targeting aging directly? And so the first thing to note is that we haven't done this yet, right? This is hypothetical in people, but in laboratory animals, it's actually quite routine to target aging directly, increase lifespan by 30%. And so if we were successful at doing that in this same 50-year-old woman, the effect on life expectancy is proportionally much greater, which is what's shown here, than curing individual diseases, with the added value that those extra years are predicted to be spent in relatively good health. And that's what the green shading is meant to illustrate. Because we have not only fixed one functional decline, one disease, we have pushed back all of the diseases and declines in function of aging simultaneously. And that's sort of referred to as this term, the longevity dividend, which Jay Olshansky coined to reflect the added value from targeting aging directly. And so that leads to the next point that I want to make, which is that in the field, we use lifespan fairly extensively as a metric for affecting aging because it's easy to measure. But the goal isn't primarily to extend lifespan. It's really to maximize this period of life spent free from chronic disease and disability, which we refer to as health span. So to push back those diseases of aging as far as possible into the later years of life. Okay, so a couple more big picture points that I want to make. One is that aging is just biology. And the reason I think this is an important point to make is that I think oftentimes when people start to talk about slowing aging, that sounds kind of like science fiction. But I want to make the point that this is science fact and this is happening. And one way to think about this is to just look at nature, right? Nature has figured out through evolution how to modulate the rates of aging by many orders of magnitude. And so if we just look across the animal kingdom, which is represented here, we can find animals that age, grow old, and die in a few months, like fruit flies, which are commonly studied in the lab, a few years, like mice in the laboratory, or a decade for some medium to large sized dogs. And then there are animals that live, exceptionally long. So whales that can live a couple hundred years, or clams that can live 500 years. And there are interesting cases like this sort of wrinkly pink guy here, the naked mole rat, where genetically the naked mole rat is relatively closely related to other rodents, but it ages about 10 times more slowly, right? So if we can understand, what are the differences between the naked mole rat and laboratory mice and rats, that tells us something about mechanisms that can quite dramatically affect the rate of aging. So aging is just biology. And if you think about what's different among these different animals, right, you can really boil it down to a couple of things. Their genetics, right? Their DNA is different, and their environment. And that's just biology. So, so nature has figured out how to dramatically alter the rates of aging. And we're starting to understand that from a scientific mechanistic perspective. And one way to appreciate that is through what are called the hallmarks of aging. And this is a figure from a review paper that was published in Cell going on 8 years ago now, called The Hallmarks of Aging. And I don't want to go through what these specific hallmarks are today. I just want to make the point that as a field, we have learned a lot about the biological mechanisms of aging. We can start to categorize these hallmarks that are conserved molecular mechanisms of aging that are shared, broadly speaking, across all mammals and the animal kingdom. And as we start to understand what these mechanisms of aging are, individually, any of these hallmarks of aging represent potential therapeutic targets that we can modify to affect aspects of the aging process. And one of the exciting things that has emerged is that at least some of the genetic pathways that we are studying and the interventions that we're studying to affect the biology of aging seem to target these many, if not all, of these hallmarks of aging simultaneously, which I would interpret as broadly slowing the aging process. So aging is just biology, and we're starting to figure it out. There's a lot to learn, but I think we've gotten far enough as a field that we can actually intervene in the aging process in a meaningful way that might have clinical application in the relatively near future. And these are just some of the approaches that are now routine in the laboratory for modifying the biological aging process. There are a variety of genetic pathways that have been studied extensively in all of the common model organisms that we use. So budding yeast, nematode C. elegans, fruit flies, mice. These pathways have been shown genetically that if you if you modulate them in the appropriate direction, you can extend lifespan. And so one of the things to appreciate is there seem to be fundamental genetic regulators of aging that are broadly shared, evolutionarily speaking, across the animal kingdom. And then there are what I call lifestyle or drug interventions that also can modulate the aging process and tend to impinge on these same genetic pathways. This is not an extensive list, but it's a partial list of the best studied interventions. And one that I'm going to focus on for the next 15 minutes or so is the mTOR pathway, the mechanistic target of rapamycin, and this drug, rapamycin, which is a specific inhibitor of mTOR. And the protein mTOR was actually named after the drug, as you might have guessed. So first, just a very little bit about the background on rapamycin and mTOR. So mTOR is a nutrient responsive kinase. So it's involved, you can sort of think of it as a key sensor that the cell uses to sense the environment and kind of make a fundamental decision. And that fundamental decision is based on the environmental conditions, is it a good time to grow and reproduce? Or is it better to shut down growth and reproduction and become stress resistant, right? And if you think about that from an evolutionary perspective, it makes a lot of sense that you would want to have a control point like this, right? And if you think about the components of the environment that are most important for this decision, one of them is going to be how much food is around, right? It's not a great evolutionary strategy to have a bunch of babies when there's no food, right, to feed them. So you can kind of think of TOR as being the sensor that detects how much food is available in the environment and helps the cell or the animal make this decision. And so TOR directly senses amino acids available in the environment. It also responds to hormones that are responsive to nutrient availability, like IGF-1 and growth hormone. And it also senses a variety of different stress signals. It integrates that information, and then through these downstream processes, which are still being worked out, this may not even be a comprehensive list, but you'll see mTOR does a lot. Through these downstream processes, it then helps the cell make this appropriate decision: grow and reproduce, or shut down growth and reproduction and become stress resistant. And I want to make a couple of points about this. One is that mTOR has been selected, I think, to make that decision. But it happens to turn out that being stress resistant is also consistent with slower aging, because when you're stress resistant, you tend to tamp down on growth, and that reduces the accumulation of these hallmarks of aging. And so what has been observed is that when we turn down mTOR signaling, either by reducing nutrients available in the environment, so caloric restriction, for example, that affects these downstream processes through mTOR, and that leads to slower aging. And it really seems to be the case that inhibition of mTOR delays many, maybe all of these hallmarks of aging that I talked about previously. And again, that can be done genetically, can be done by genetically turning down mTOR. It can be done environmentally by reducing amino acids or by caloric restriction. And it can be done pharmacologically with this drug rapamycin. And the observation is, in all of the model organisms that I showed you before, yeast and worms and flies and mice, when you do that, you slow aging, you increase lifespan, and appear to delay the functional declines and these hallmarks of aging that go along with the aging process. So there's just a few additional things to mention about rapamycin, the drug. So it's actually a very interesting backstory to this drug. It was identified from soil samples on Rapa Nui or Easter Island. That's where the drug gets its name. It's a specific inhibitor of mTOR complex 1, which I just talked about. And in fact, from a biochemical perspective, rapamycin is an extremely clean drug. As far as we know, all of the effects that are seen from rapamycin come through this mechanism of inhibiting mTOR complex 1. It is the most effective longevity drug to date, routinely increases lifespan up to about 30% in mice. One of the interesting and important aspects of the studies on rapamycin so far in the context of aging in mice is you get pretty much the same effect on lifespan if you start in middle age. So this was work that was originally done by the NIA Interventions Testing Program, where they waited until the mice were 20 months of age, which is about the mouse equivalent of a 60, 65-year-old person, and started the treatment then. And they still saw this robust increase in lifespan. And then, as I alluded to earlier, what we're really interested in is health span more so than lifespan. And rapamycin seems to broadly delay declines in function associated with many different tissues, including brain, heart, immune system, the list goes on and on. So it really seems to be broadly affecting the aging process, not only affecting lifespan in mice. And what I'll tell you in a minute is that and this is one of the most exciting things for me to emerge over the last several years. Not only does it seem to be the case that rapamycin delays these declines in function, but we can actually see reversals of functional decline after they've already happened. And I'll show you 3 examples of that in a minute. So based on all of that, you might ask yourself, why aren't we taking rapamycin, or where can I get rapamycin? And I think there are a variety of reasons. One being we haven't yet proven that rapamycin affects aging in people, although there's some reason to believe that's the case. But also, when you look at the way rapamycin has been used clinically, so it is FDA approved, it's off patent. There's a generic version called Rapamycin or sirolimus. It has sort of a bad reputation clinically. And that's mostly because of how the drug has been used historically. So it was FDA approved to prevent organ transplant rejection in patients who have had a mostly kidney transplant, but also other organ transplants. And so in that context, the drug has been used at very high doses in combination with true immunosuppressants. And so in that context, there is a long list of side effects associated with the use of rapamycin and rapamycin derivatives. This is a short list here. But for that reason, the drug has sort of developed a bad reputation among clinicians. And those side effects are real, no question about it. I think it's important to appreciate, though, that these are sick people taking high doses. And there is a growing body of, at least in my view, quite convincing literature that in healthier people, and in some cases, people without any significant age-related disease, taking lower doses of rapamycin, usually as a monotherapy, there are very few side effects. And in fact, it's a quite safe drug. And I would actually say that, in my opinion, there is good reason to believe that rapamycin is as safe, if not more safe, than aspirin at low doses in healthy people. There are clinicians who would disagree with that, but I would say I know the data better than they do. So I think we will discover over time that the bad reputation for rapamycin is largely undeserved. Nonetheless, it's there. And I think that's a barrier to clinicians really thinking seriously about translational use of rapamycin for age-related indications in healthy people. And so we set out to ask whether or not you might be able to get some of these benefits of rapamycin from short-term treatments. The idea being that transient treatments with a drug are less likely to have significant side effects and are more likely to be viewed as safer by the clinical community. And so this is now work that's a few years old, where we did a very simple experiment. We took mice, laboratory mice, we aged them to 20 months of age, and then we treated them with either a control or with rapamycin for 3 months, so for 12 weeks. And then we stopped the treatment and we just put them back on the shelf and waited and saw how long did the mice live. So again, these mice in the control group and the rapamycin treated group are treated identically up to 20 months of age, and then from 23 months of age for the rest of their life. It's only that 3-month window that there's a difference. And this is the result of one of the cohorts. This is published, so I'll refer you to the paper if you really want to dig into the data. There's actually quite a bit of interesting data, I think, in that paper. But this is one of the cohorts where the 3-month treatment of rapamycin led to a 60% increase in life expectancy. So this is now showing you survival after the end of that 3-month treatment period. And so there's a lot of interesting things to take from that. But one of the things that I find most interesting is that this transient treatment with rapamycin of 90 days led to an absolute lifespan extension of 141 days in this group, which means that the magnitude of effect was actually bigger than the length of time the animals saw the drug. And when you think about potential mechanisms for how you would get a bigger lifespan extension than the length of time the animals got the drug, it's kind of interesting to think about what that means. And I'll kind of allude to what I think might be going on here as I go through my talk, but it was an interesting observation. And if we look back to that 50-year-old woman that I mentioned hypothetically towards the beginning of my talk, again, we don't know that this is going to work the same way in people, but if it did, a straight linear extrapolation, that would be about 2 decades of life expectancy increase for that 50-year-old woman that I talked about earlier. So we're talking potentially quite significant effects on life expectancy. Okay, so that's lifespan, which is interesting and it's useful because it's easy to measure. What about health span? And so I'm going to give you 3 examples where transient treatments with rapamycin appear to improve functional measures of health in 3 very different tissues. The first one is heart. So this is data from Peter Rabinovich's lab at the University of Washington. There's also data from Simon Melov's lab at the Buck Institute that is very similar. And so this is a similar sort of design to that experiment I just showed you that we did, where they had either young mice or old mice, the old mice being 20 months of age. They treated them with either control or rapamycin for 10 weeks. And then they looked at measures of left ventricular function, so that you can just think of these measures as how well is that chamber of the heart working? How effectively is it pumping blood with each contraction? And so there's 2 measures that are shown here. One is LVMI, or left ventricular mass index, and the other is E to A ratio, which is really a measure of the dimensions of that chamber with each contraction and how it changes. So with age, the other thing to note is the open bars are before the 10-week period, and the black bars are after the 10-week period. So first thing to note with LVMI is that LVMI increases with age. If you just look at the open bars going from young to old, there is an increase in LVMI with age. And before the treatment, the mice that are destined to get rapamycin, they haven't gotten it yet, have essentially the same LVMI as the old mice that are destined to get the control. So then what happens after the treatment period? No change in the mice that get the control. That's what you would expect. The mice that got rapamycin, their LVMI is reduced to a level that is not significantly different than the young animals. So at least for this measure, it looks like 10 weeks of rapamycin is suppressing the age-related change in LVMI. And if you look at E to A ratio, a very sort of similar outcome, except in this case, E to A ratio is decreasing. So the function of the heart, of that chamber of the heart, is decreasing with age. It actually continues to decrease in the mice that get the control over the 10 weeks. That kind of makes sense. This is the point in life where function is really sort of dropping off the cliff. But in the mice that got rapamycin, that measure of heart function is actually all the way back up to the young mice. So again, for this particular measure of cardiac function, 10 weeks of rapamycin treatment is taking that old heart and making it function like a young heart. This is a study of immune function in mice from Pan Zheng's lab at Michigan. Again, a similar design. In this case, the mice are 24 months of age before they get either 6 weeks of control or 6 weeks of rapamycin. And then the question is, what effect does that have on immune function? And in this case, they used response to an influenza vaccine as their readout. So after the 6 weeks with either the control or rapamycin, the mice get a flu vaccine. And then they wait 2 weeks, and then they're challenged with what would be a lethal dose of influenza in an unvaccinated mouse, okay? And the question is, if the vaccine worked, are those mice protected from that lethal challenge with influenza? And this is the survival data following this experiment. So the first thing to note is the young mice that don't get immunized. That's the solid black line here, okay? This is just showing that indeed, in a non-immunized state, this is a lethal challenge with influenza. So all of the mice die within about a week if they're not immunized. If you're an old mouse and you don't get rapamycin, but you do get the vaccine, and this really was striking to me the first time I saw this, there's about a 2/3 chance that the old immune system will not respond to that vaccine. And when challenged with influenza, then the mice will die. And then there's about a third of the mice where the immune system does what it's supposed to, the vaccine is effective, and the mice are protected against that influenza challenge. So that's pretty striking. If you're a young mouse and you get immunized, there's 100% protection. That's what you would expect for a young immune system. It's functioning exactly like it's supposed to. So this is the first thing, right? There's a clear age-dependent decline in immune function in mice as measured by response to a flu vaccine. And there's pretty good evidence that something similar happens in people. Now, if you're an old mouse that got rapamycin for 6 weeks, there's a dotted line here under the solid line. 100% Vaccine response, 100% protection. So again, at least for this particular measure of immune function, 6 weeks of rapamycin treatment is enough to fully restore the aged immune system functionally back to that of a young immune system. And I think this is particularly interesting to think about in the context of human immunity for a couple of reasons. One is now there have been 2 clinical trials. These are both phase 2 clinical trials with a derivative of rapamycin, biochemically functions exactly the same way, suggesting that in healthy older people, this derivative of rapamycin, 6 weeks of treatment can improve flu vaccine response and also protect those people against other types of respiratory tract infections over the next year. So again, it's a little bit, it's not quite definitive, but it's pretty close, suggesting that at least for this particular immune outcome, rapamycin probably can functionally rejuvenate the immune system in healthy older people. And given that we are all still living through this global pandemic, this is important, I think. We know that COVID-19 is a disease of aging. I think it's not as appreciated as it should be, but it absolutely is the case that the older you are, if you get infected, your risk of hospitalization and death goes up exponentially, just like all of these other age-related diseases that we think a lot about. As I told you, rapamycin can rejuvenate immune function in mice, probably can rejuvenate immune function in people. And so I think there's at least the possibility that rapamycin could have an effect on this age-related risk profile for severe outcomes from COVID-19. And this is complete speculation, but if we just go based on what we see in mice, it might take the outcome response from what it was to something like this, where we can dramatically reduce the risk of severe outcomes and death in the elderly. And I think, obviously, for good reason, there's a lot of enthusiasm and excitement about COVID-19 vaccines. But I think we need to recognize that it's likely, at least for most of the vaccines, that it's going to be very much like with the influenza vaccine, that the people who need it the most are the least likely to actually get the benefits from the vaccine. And so again, it's possible that rapamycin or another mTOR inhibitor could enhance the ability of the elderly immune system to respond to COVID-19 vaccines. Okay, so I showed you heart, I showed you immune system. Now I want to do a little bit of a deeper dive on an area that my lab has been working on, which is the effects of rapamycin on aging in the oral cavity. And this is really work that was started in my lab by Jonathan Ahn, who was a dental PhD student working in my lab and is now an assistant professor in the Department of Oral Health Sciences at the University of Washington. And so John came to me 6-7 years ago now and made me think about something I had never thought about before, which is that periodontal disease is a disease of aging, just like Alzheimer's disease, just like diabetes, just like cardiovascular disease. If you look at the statistics, the risk of developing periodontal disease goes up with age, just like all of those other diseases. And it's actually quite prevalent. So in the United States, something like 2/3 of people over the age of 65 will have periodontal disease. And so John presented that to me, and we started thinking about questions of, could we model that in laboratory mice? And then if we could, is it the case that the same mechanisms that we think affect biological aging in other tissues also affect periodontal disease in mice? And so John set out to model this in mice. He showed that indeed, with age, mice develop the clinical features of periodontal disease, so gingival inflammation, loss of bone around the teeth, and pathogenic dysregulation of the microbiome, and that 8 weeks of rapamycin could suppress all 3 of those phenotypes. And that's published in eLife. I'll show you a little bit of the data, but if you're interested, I'll refer you to the paper. And this is just showing you one of the kinds of data that we got from this. These are micro CTs of the bone, which is shown here around the teeth. And it was actually quite striking in some of these animals. These are 20-month-old mice. So this is before the rapamycin treatment. You can see there are these spaces here where the bone has receded towards the root of the tooth. You can actually see the space here. We could actually see within 8 weeks regrowth of bone to fill in that space in exactly the same mice. So this is a really nice type of data to actually convince us that rapamycin was having this pretty striking effect on a bone around the teeth. So this is sort of the design of the first first wave of experiments that we did, which was to take either young mice, there's actually an intermediate group, and adult mice, which was 12 months, or aged mice, do this 8-week period, and then do micro CT scans like I just showed you before and after that 8-week period. And then in the old mice, we had either control, which is Eudragit. You might see this referred to Eudragit on the subsequent slides. That's just the encapsulation around the rapamycin, but with no rapamycin, or rapamycin at this dose, 42 parts per million, which is a dose that's commonly used in the field. So we did this experiment and we looked at these 3 clinically defining features of periodontal disease. And as I mentioned, we saw evidence that for reversal for all 3 of these clinically defining features of periodontal disease. And I just want to show you a little bit of data here because it'll be relevant for some of the unpublished data I'm going to show you. One is looking at bone homeostasis. So I told you that we saw regrowth of bone, and we wanted to drill a little bit deeper and ask, could we see evidence for changes in factors that are known to regulate either formation of new bone or reabsorption of bone? And so we looked at 2 factors, rankle and OPG. And so you can think of this as rankle is promoting bone loss and OPG is promoting bone formation, okay? And they affect, they're markers of osteoclast and osteoblast function, essentially. And so what John was able to show was that in the periodontal bone with age, there is a pretty dramatic increase in rankle expression that's suppressed by rapamycin. And if we look at the ratio of rankle to OPG, so again, higher numbers here are promoting bone loss, at least that's the prediction. And that fits what we see by micro CT, that there's an increase with age, and that's suppressed by 8 weeks of rapamycin treatment. So we're seeing the same thing we expect to see with these markers of osteoblast and osteoclast function. And then we looked at inflammation, because as I told you, inflammation is a clinically defining feature of periodontal disease. And he looked at a variety of different types of inflammation, and NF-kappa B mediated inflammation in particular gave a very strong signal. So NF-kappa B is a transcription factor that drives inflammatory response in many tissues. It's known to increase with age in other tissues. So we looked at a couple of different measures of NF-kappa B activity. We looked at the abundance of this activating subunit, P65, as well as abundance of the inhibitory subunit, I-kappa B, and phosphorylation of the inhibitory subunit. So the way you can think about this is that more P65 means more active NF-kappa B, more inflammation. That's a simplification, but it'll work for today's purposes. More I-kappa B would be inhibition of P65, lower inflammation. And then phosphorylation of I-kappa B is a post-translational modification that promotes degradation of I-kappa B. And so it's an inhibitor of an inhibitor is a way to think about it. So more phosphorylation of I-kappa B means more activity, higher inflammation. So in the gingival tissue, so the gums, the gum tissue, with age, there's a pretty dramatic increase in P65, so activation of NF-kappa B, and a decrease in this inhibitory subunit. Both of those are suppressed by rapamycin. So prediction of enhanced inflammation that is now being suppressed by rapamycin in the gums, that fits with periodontal disease. And in the periodontal bone, it's a little bit different. We don't see substantial changes in amount of I-kappa B, but we see pretty robust changes in phosphorylation of I-kappa B that go the direction we would expect. So increased phosphorylation of I-kappa B with age means less activity of that subunit, more inflammation, and that also is suppressed by rapamycin. Okay, so and then we looked at actually cytokine abundance, so targets of NF-kappa B in both the gum and the periodontal disease. And so this is just showing you the outcome of that experiment. This is now fold change relative to young. So anything above this dotted line here means there was an increase in the abundance of that cytokine in the control animals in pink, or the control animals that got rapamycin, the old animals that got rapamycin in blue. What I hope you can appreciate is that for a variety of inflammatory cytokines, there's an age-related increase, and rapamycin suppressed both of those. And this was really interesting to us because if you know anything about inflammaging, or the increase in inflammatory signaling that goes along with aging, you will appreciate that these cytokines represent what people think of as the SASP, or the senescence-associated secretory phenotype. So with aging, there's an accumulation of senescent cells. They give off these inflammatory cytokines, and that's thought to drive a lot of the chronic inflammation that goes along with aging. So this made us wonder whether there might be a link between accumulation of senescent cells in the oral tissue and the effects that we were seeing with aging and with rapamycin. And so we've done a little bit of preliminary studies looking at this. We looked at P16-INK4A, which is a marker of senescent cells. Indeed, there is a dramatic age-related increase in P16 abundance with age in both the gum and the periodontal bone. And this was really striking to us, is completely suppressed by 8 weeks of rapamycin treatment. So this is now unpublished data. So we're very interested in understanding, is this really a senescence-mediated phenomenon that we're seeing? The other question that we've been asking is, these effects that we see with 8 weeks of rapamycin, do they last? In other words, after you stop the treatment, do they go away? Or do they persist for a while? And so to address that, we've done sort of a crossover design here where we have these old mice. They get either control or rapamycin for 8 weeks. And then after that 8-week period, the control mice get rapamycin and the rapamycin mice get the control. And we can ask, do we still see the same effects from rapamycin here? And in the mice that go back on the control, do we lose those effects from rapamycin? And we're just starting to look at this data. This is really work that John is doing right now. But at least for a couple of the key measures, it looks like the effects of rapamycin are persistent. So if we look at the bone markers, so rankle, again, age-related increase of rankle, Rapa, 8 weeks suppresses it. The mice that got the control and then went on rapamycin for 8 weeks, you can see it worked in those mice. And the mice that got rapamycin for 8 weeks and then went back on the control for 8 weeks, it's still reduced. So for this measure, the effects of rapamycin persist, again, at least for 8 weeks, maybe for longer. And also for the NF-kappa B marker, I-kappa B phosphorylation, dramatic decrease with rapamycin with age, and that persists in both groups of this crossover. The ones that get rapamycin for 8 weeks are the ones that go back on the control for 8 weeks. So at least for 8 weeks, the effects of rapamycin seem persistent for these two measures. And that also seems to be true for the bone loss. And so now this, I haven't yet told you exactly how we're measuring bone loss in these mice. The way this is done is by micro CT, and it's by landmarking the somato-enamel junction and the alveolar bone crest. And so the alveolar bone crest is really where the periodontal bone is at. How high does it go? And with age, as bone is lost, this alveolar bone crest recedes. And so you can see, just going from a young mouse to an old mouse, the distance between these landmarks gets bigger. And so that's how we're measuring periodontal bone loss. So a higher number here, somato-enamel junction minus alveolar bone crest, means less bone, okay? So in old mice at baseline, this is showing you now 8 different animals. You can see there's really no difference in the bone levels in the 8 weeks on control for each individual mouse. But in every mouse, to different extents, but in every mouse, when they go on rapamycin, there's regrowth of bone. In other words, this distance is getting smaller. And so that's pretty striking that we can actually see that in these animals over this 8-week period. And then you can see the same thing for the mice that get 8 weeks of rapamycin and then go back on the control diet. Those changes, that increase in bone, persists when they go back on the control diet. And in some cases, it looks like the bone continues to regrow. So there might actually be continued benefit from that short-term period of rapamycin, even after the rapamycin is taken away. Okay, so this obviously opens up lots of questions. We don't have enough time, unfortunately, to do everything we want to do, but we're trying to address some of the questions that I think are most interesting. One question is, so we're growing new bone, that's great. Is it good bone, right? You could imagine that maybe we're growing really frail bone, which wouldn't be fantastic. We've got a little bit of data, and in fact, it does look like the bone is quite strong. So it looks like it's just as strong as younger bone. But we still need to really nail that down. We're also looking in other oral tissues, and at least for the salivary gland, we see very similar data that it looks like rapamycin is reducing inflammation, restoring function. So that's really exciting. Dry mouth is actually a big problem for a lot of elderly people. So we think that this may be more broad than just the gingival tissue and the periodontal bone. We're asking, is this mediated through senescent cells by treating mice with some drugs that are thought to specifically clear senescent cells. We don't have the data for that yet, but that experiment is in progress. Another interesting question is, does this require systemic inhibition of mTOR? Or if we were able to give rapamycin only in the gums, would we be able to get these effects? And I can think of models that either result could be true. If this is mediated, at least in part, through effects on the systemic immune system, we might need systemic delivery of rapamycin. So we're hoping that we'll be able to address that by specifically dosing the gingival tissue and see if we can recapitulate these effects in mice. And then the big question is, will this work in people? And so we have not yet started a clinical trial, but we're hopeful that we'll be able to do a clinical trial to actually address that. And I really like periodontal disease as an endpoint. If you think about age-related endpoints, it's hard to imagine one that's easier to look at than periodontal disease. Dentists are quite good at diagnosing inflammation in the gum, at measuring bone levels, both by pocket depth and by X-ray. So I think this is amenable to a clinical trial that could give pretty robust readouts if there's an effect. So we're hopeful that we'll be able to do that at some point. Okay, so now I'm going to shift gears just for the last few minutes and tell you a little bit about the dog aging project. So this was really brought about from this question, at least in my mind, for, we've been quite successful at targeting aging in laboratory animals, but we really don't have a great strategy for bridging the gap between laboratory animals and people. And I'm certainly happy to talk with anyone who's interested about some of the challenges of doing a clinical trial for healthy aging in humans. It's challenging, as you can imagine. So one approach that we have explored is it possible that our companion animals, and pet dogs in particular, can kind of serve as a bridge between laboratory studies and the human environment? And I have a double-headed arrow here because I absolutely believe that this is partly about what we can learn about human aging, but it's also largely about what we can learn about aging in pet dogs and hopefully increasing the healthy longevity of our pets. Because I view clearly there's intrinsic value if we can help people's pets live longer, healthier lives. So there's really two major components to the dog aging project. One is a large-scale longitudinal study of aging, and the goal of that is really to define the genetic and environmental factors that influence healthy aging in pet dogs. And then the second part of the dog aging project is to actually do something about it, to ask whether or not these interventions that seem to slow aging in laboratory animals can have a similar effect in pet dogs. And the first intervention we're starting with is rapamycin. So why dogs? Why pet dogs in particular? And I think there are lots of reasons. These are just a few of the big ones. One is that dogs age very much like we do, but they do it faster. So we're all familiar with the idea that 1 human year is about 7 dog years. That's obviously not perfect biologically, but it's pretty good. And all that really means is dogs age about 7 times faster than people do. They get most of the same age-related diseases that we do. They show most of the same functional declines that we do. So that means that we can actually study aging in a reasonable time frame in dogs. Dogs have this very unique genetic and phenotypic diversity. So there are a couple hundred purebred breeds of dogs, which are, you can kind of think of as inbred strains. But on top of that, we've got this mixed breed population. And so from a genetic perspective, that's really powerful to have that sort of genetic architecture combined with the fact that dogs have phenotypic diversity that really exceeds that of people in almost any trait that you think of. Just think of body size and the difference between a Chihuahua and a Great Dane. So genetically, dogs are quite powerful for mapping genetic variants that are associated with traits. And this is a really important one because I think it's something that in the aging field, we really cannot model any other way. Dogs share the human environment more than any other animal, maybe with the exception of pet cats. And really with the exception of diet, dogs share every aspect of the human environment. And so we can understand aging in that context, which is something we really can't do in laboratory animals. People love their dogs, and this is a citizen science project in every aspect of that term. Our owners are engaged and active. And so I think this engages the public in science in a way that few projects can. And as I mentioned, there's intrinsic value to improving the quality of life for pets and owners. And these images here are sort of meant to represent the environmental diversity. I don't know if you can see this on your screen, but this is a dog. This is the dog house. So just like in people, there are rich dogs, there are poor dogs, there are dogs with healthier lifestyle habits, and there are dogs that maybe don't have a particularly healthy lifestyle. I make this disclaimer every time I show this picture with the dog with the cigarette in its mouth. I had nothing to do with that picture. I did not pose that dog. This was just came off the web. The dogs obviously don't smoke, but they do breathe the same air that we breathe, and they drink tap water by and large. And so we can learn a lot about environmental factors that are associated with health outcomes during aging from studying aging in dogs. This is the structure of the Dog Aging Project as it currently stands. We have the largest component of the Dog Aging Project, which we call the Dog Aging Project PAC. And to be part of the Dog Aging Project PAC, owners nominate their dog through the website and complete a large, what we call health and life experiences survey. This is a fairly extensive survey. It takes a couple of hours to complete that captures as much information as we can about the dog's health history and its environment. Everybody who does that is part of the Dog Aging Project PAC, and they are asked to send us their veterinary electronic medical records. We hope that we'll get the PAC up to 100,000. Right now it's at about 30,000. We have about 30,000 dogs enrolled in the PAC at this point. Within the PAC, then, a subset of dogs are selected for the foundation group. They get their genome sequenced, 10,000 dogs in the foundation. Then there's the omics group, the precision group, which in addition to genome sequencing every year, we will get blood metabolome, microbiome, fecal microbiome, and blood epigenome. And those dogs will go into the clinic every year and will get, of course, extensive veterinary records. And then there's the centenarian group, which are just dogs that live longer than they are expected to. They get high resolution genome sequencing. And then there's the clinical trial group, which I'll tell you a little bit more about. This is a double-blind, placebo-controlled clinical trial of rapamycin that we call Triad for test of rapamycin in aging dogs. Okay, so as I mentioned now, we're up to almost 30,000 dogs in the PAC. This is our 25,000th member, Sage. Sage lives in New Hampshire. And every dog in the PAC, we have the health history, electronic medical records, owner demographic, dog demographics, and then a bunch of information on lifestyle, and then the geographic data based on where they live. And this is showing the distribution around the United States of where the PAC members are. So we're just now starting to get into the data. So I'm just going to show you a very sort of tip of the iceberg view of what the data looks like. This is all from the owner survey. So we don't yet have the genome sequencing, the omics. That'll be coming in the next year. But still, just from the owner survey, there's a lot of cool information. This is showing you owner-reported disease diagnoses. You can see we have a lot of disease diagnoses in the dogs in the PAC. Skin disorders are number one, but there's a lot of age-related diseases. I talked about oral disease, a lot of oral disease in the dogs, a lot of eye disease, of course, a lot of cancer and cardiac disease. So there's a lot of information here for us to dig into on age-related disease incidents. And we can see, you know, in some ways what we expect, but it's nice to see this come out of the owner-reported survey data. So just like in people, average walking speed of dogs declines with age. That makes sense. Kidney disease increases with age. This is a log scale. So again, just like in people, there's essentially a logarithmic increase in kidney disease with age. We can break this out by body size. So big dogs tend to age faster than small dogs. And we can also break it out by sex. And at least for kidney disease, there's this interesting observation that within all body sizes, female dogs seem to be at higher risk age matched than male dogs. We can look at cancer. Again, logarithmic increase in cancer incidence with age, all owner reported. And here there's a dramatic size effect. So at every age, big dogs are much more likely to be diagnosed with cancer than small dogs are. Again, not super surprising, but it's nice to see such strong correlation come out of the owner reported survey data. And one of the interesting things we can ask is about multimorbidity. So first of all, do dogs develop multiple age-related diseases with age, as people do, multimorbidity, having more than one? And if so, which go together? And this is really interesting because this is very early, so I don't want to make any conclusions about the multimorbidity network so far. But I think as we get all 30,000 records analyzed, we'll be able to come up with a pretty interesting network view of which diseases go together as dogs are aging, which is something that I think will be informative maybe about interconnected mechanisms of age-related diseases. This is something that I'm interested in. So those of you who have followed the aging field even at a distance will be familiar with the idea that intermittent fasting and time-restricted feeding have sort of caught the popular attention as ways to slow aging without a ton of evidence that they actually work in people. But in mice, there's some evidence that you can affect age-related outcomes from intermittent fasting or time-restricted feedings. Dogs might actually be a really interesting way to model this in a more natural environment, because some dogs are fed all the time. This is just showing you our owner-reported meal frequency. Some dogs are fed 3 times a day. The majority of dogs are fed 2 times a day, but about 10% of dogs get 1 meal a day, which would actually be pretty close to what people think of as time-restricted feeding in mice. And so we can ask, are there associations between meal frequency and health outcomes of aging in dogs? We haven't done it yet, but I'm pretty excited to see what that looks like. Okay, so that's the longitudinal study, and I know I'm running a little bit over, so I will just very quickly give you an update on the rapamycin clinical trial. This is the test of rapamycin in aging dogs, or TRIAD. The goal here is really to answer the question, does rapamycin slow aging in companion dogs? I already told you about the rationale for why we think that's a question that needs to be answered. And as preliminary data, we've completed a 10-week randomized clinical trial, mostly for safety. That's published. It was published in Geroscience in 2019, so you can check out that paper if you're interested. The preliminary data there looked like we were seeing similar improvements in heart function as I showed you from Peter Rabinovich's lab in mice. So at least it was consistent with the idea that rapamycin could be tested safely in pet dogs and might have effects on age-related heart function. So because this is a study in people's pets, safety always has to be at the top of our minds. And so we have designed all of our clinical trials with safety first in mind. We have an animal welfare advisory board, which is populated by members of the public and veterinarians who are interested in this topic, as well as an NIH mandated data safety monitoring board. This is a true double-blind, placebo-controlled randomized clinical trial. It's open to all genetic backgrounds. The dogs have to be middle-aged, so at least 7 years old, and they have to be medium to large size, at least 40 pounds. And that's, again, because big dogs age faster than small dogs. And all of the dogs are seen at one of our 7 partner veterinary teaching hospitals, which are shown here. So for the TRIAD study, all of the clinical component will be at one of these 7 clinical sites. This is the design. I'll just very quickly take you through it. So the dogs are pre-screened. This is a healthy aging study, so they can't have any pre-existing significant age-related conditions. So some dogs will be excluded at that point in the study. Our goal is to randomize 350 dogs equally between placebo and rapamycin treatment group. The dogs in the rapamycin treatment group will receive rapamycin for 1 year and then 2 years of follow-up. And the clinical visits will be every 6 months. So baseline and every 6 months after that. At every clinical visit, we will get echocardiograms to look at heart function. We'll get blood metabolome, blood epigenome, blood chemistry, fecal microbiome, cognitive assessments, and there will be activity monitoring. And then, of course, we'll track disease incidence. So lifespan is the primary endpoint. We are powered to detect a 15% change in lifespan, but age-related health outcomes, multiple age-related health outcomes will be secondary endpoints. We're using an enteric-coated version of rapamycin. The dose is once a week, 0.15 milligrams per kilogram. And our goal is to have 100 dogs screened by the end of this year, with the rest of the dogs randomized by the first half of next year. Okay, so again, just to summarize, we're now getting our first longitudinal data. So the first dogs in the pack are now filling out their 1-year anniversary data. So we have cross-sectional data on almost 30,000 dogs, and now we are a true longitudinal study because some of those dogs are now turning in their first year updates. We've rolled out cognitive assessments to all 28,000 dogs, and we hope to have half of the foundation and precision dogs enrolled by the end of this year. Actually, we hope to have all of those dogs enrolled by the end of this year. Some of them are enrolled now, and as I mentioned, 100 dogs in TRIAD by the end of the year, and we want to get the pack up to 50,000. So my call to you is, if you have a dog and you want to participate, you can go to our website. There's a little button that says nominate your dog. press that, and you will be invited to complete the Comprehensive Health and Life Experiences Survey. So please do that if you're interested. And I just want to thank the members of the Dog Aging Project team. It is a huge, awesome collaborative team from multiple institutions, too many people to recognize by name right now, but I especially want to mention Kate Creevy, who is our Chief Veterinary Officer at Texas A&M, and Daniel Promislo, who is the co-director with me of the Dog Aging Project and the PI on an NIH U19 grant, which funds much of the work that I've told you about today. And so with that, I will stop. I apologize for going a little bit over, but I'm happy to stick around for as long as people want to ask questions and answer any questions that you might have. So thank you for your attention.
Okay, well, thank you very much, Dr. Kaeberlein. Now the floor is open for the questions. I know the Matt is doing something very unique, very interesting. Okay, I'm really excited about that because I'm also involved in using the dog model as studying the human ALS, especially for the canine degenerative myelopathy. I mean, I mentioned very briefly to Matt some times ago when we were in the meeting. And this is really very, very interesting model in between from the laboratory animals and then a human. So that is a great potential. Okay, just ask about anything about the aging. I'm sure that Matt will have an answer. Please do so.
Hi, Baki. I have a question.
Yes.
First of all, thank you for an excellent talk. I truly appreciate it. I'm a dog lover myself. Nikki is my German Shepherd. Maybe I will enroll her in such a study. But my question is focused on, mTOR is a kinase, and there's such a thing as phosphorylation-dependent SUMOylation. So my question to you is, does mTOR interface with SUMO and or acetylases such as sirtuins?
Yeah, I mean, it's a good question. So first of all, I have a German Shepherd as well. I love my German Shepherd. His name is Davi. German Shepherds are the best. No offense, but that's my opinion at least. So your question, I mean, it's a good one. I think absolutely there are connections. I mean, like with any kinase, there are a plethora of potential substrates, some of which are known, some of which aren't, and some of which may be interfacing with these other post-translational modifications. And absolutely, that's going to be the case with mTOR. I don't know so much about the data on SUMOylation of mTOR substrates. With respect to sirtuins, there are definitely connections, certainly indirect connections. So it is absolutely the case that AMP kinase impinges on both mTOR activity and sirtuin activity, and so they are absolutely connected that way. That if you get changes in mTOR, you can get effects on AMP kinase and vice versa that are transmitted to sirtuins that way. And then we have some unpublished data in my lab that with respect to mitochondrial function, there is a very interesting genetic connection. And so I won't get into the weeds here other than to say that, we have published many years ago that in a mouse model of mitochondrial dysfunction, that inhibition of mTOR with rapamycin can greatly suppress the disease phenotypes associated with that model. And that was published in Science, I think, in 2013. And we've been working to try to understand the mechanisms there. And we don't understand the mechanism yet, but it seems to be the case that effect of rapamycin, at least on survival in that mouse model, is completely dependent on SIRT3, which is the mitochondrial sirtuin deacetylase. We'd have no idea. We would like to know if that's true in aging as well. I think it's an interesting connection. So it's a purely sort of genetic. We don't understand the mechanism, but you knock out SIRT3 and the effect of rapamycin in that model goes away. So there is clearly a connection. So I think the answer to your question is absolutely, but I don't understand what those connections are at this point, but they're going to be interesting to try to tease out.
I put a picture of Nikki Dog in the chat. Dr. Schneider, I see you have a question. Go ahead.
Yeah, thank you for a great presentation. So we're all standing on the same background. We understand mechanism we can apply. I can explain which results we received on immunological studies. That is kind of we all the time can look at mechanism and explain. But how we can explain there is a social aging. There is a specific North American ant insects which are produced from the same. They can produce from non-fertilized eggs. Technically, it's all twin brothers and all like queen. All right, but queen lives 2 times longer than just working. Do we have any analogy in the human life that we can apply social prevention for social aging?
Yeah, so I don't know of any analogy in people. I think mechanistically there are a couple things to consider about that particular model where you have genetically identical individuals, right? But somehow the queen can age or live substantially longer than the workers, right? So one question is the short lifespan of the workers really aging, or do they die from some programmed process, right? So that's one question. We don't know. I mean, we don't know that the queen is actually aging. It's like saying, well, I got a lot of echo there. It's like saying if at a teenage, a bunch of 18-year-olds and people die from war, they didn't age prematurely, but they didn't live as long as people who didn't go to war. So that's one question that I think has to be answered. The other model that I would say most people would point to is there's probably epigenetic changes that are really driving that. If it is really a dramatic decrease in the rate of aging, it's probably epigenetic. It's well, almost by definition epigenetically driven. But we know that post-translational modifications of histones and DNA can in fact have pretty profound effects on the rate of aging in laboratory animals. It could be extension of those mechanisms to a much larger effect size that's driving that difference. And in fact, there is a variety of work now showing that you can see transgenerational effects on aging through epigenetic modifications, where you can make these epigenetic modifications in one generation and they have persistent effects on lifespan, for 3, 4, 5 generations. That's been studied quite a bit in C. elegans, actually, where you can actually see these epigenetic transgenerational effects on aging. So that would be my first guess as to what the mechanism would be. But I don't know of any evidence in people. I mean, certainly there's evidence that there are epigenetic persistent effects on health outcomes during aging in people, but not of that magnitude, right? Where we're talking 10-fold differences in life expectancy.
Thank you so much.
I see Dr. Wolf's hand is up.
I was going to comment on that a little bit as well. I mean, 20 years ago, I had people point out that some of my high school classmates that didn't go to college looked 20 years older than I did. And I know Dr. Schneider has pointed out the benefits of college education along the way. So that's interesting to me. But the question that I end up with is, how is a person supposed to die? How does a healthy age person die? And do we in fact save money by prolonging the amount of time that everybody stays alive?
Yeah, I mean, I think that's really important. Obviously, I'm not an economist, so I can only tell you what I've been told. There are economists who have looked at this question. J.L. Shansky and colleagues have actually done, I think, a pretty comprehensive analysis, and they actually need to do another one because they themselves say that they underestimated the economic impact of healthy aging. It obviously depends on social forces that are hard to predict. But if you make the assumption that people living longer maintain function and are able to continue to productively contribute to society, the economics are pretty clear that there is a huge economic value to doing that. Now, in order for that to happen, there would have to be changes to Social Security, age, and all of that stuff, right? Which is complicated and beyond it's above my pay grade, frankly. But I think you can make the economic case that if we're successful at pushing the diseases of aging back, maintaining functionality later in life, and the compressing morbidity, so reducing the period of life that people are kept alive with one or more age-related disease, there is huge economic value in that. And in fact, you can also make the case that what we've done over the last 50, 60 years, we have improved life expectancy, but if you make the assumption that people living longer maintain function and are able to continue to productively contribute to society, the economics are pretty clear that there is a huge economic value to doing that. Now, in order for that to happen, there would have to be changes to Social Security, age, and all of that stuff, right? Which is complicated and beyond my, it's above my pay grade, frankly. But I think you can make the economic case that if we're successful at pushing the diseases of aging back, maintaining functionality later in life, there is huge economic value in that. And in fact, you can also make the case that what we've done over the last 50, 60 years, we have improved life expectancy, but people are living longer by and large with one or more age-related diseases, which is quite expensive, right, from a healthcare perspective. So we haven't done a great job at pushing diseases of aging back. We've done a great job at keeping people alive with one or more age-related diseases.
I see Dr. Agbas.
Yeah, well, I have a recipe for Dr. Wolf. My father jokingly used to say, how do people die? 3 days in the bed, 4th day you are in the grave. So, Matt, I have a question. It's anecdotal. Some of the people living in the Caucasian mountains, they live longer. This is a kind of isolated community, not much this industrialized life. And they are in the high altitude. And then they live longer. I mean, well over the, I don't know, if the newspaper is correct, something more than 120 years or something like that. I mean, when you mentioned about the epigenetical or environmental contributions to the healthy living and the long age, it just popped in my mind. Can you comment on that one?
Sure. Yeah. So, I mean, I think that specific case, right, there are these hot zones or whatever where people appear to live longer than you would expect based on the surrounding areas. And, you know, the first thing I'll say is oftentimes it's difficult to really document how big that effect is. So you have to accept that sometimes it's a little bit squishy, but it definitely is the case that, you know, there are these places where it appears that people are healthier and live longer. And hypoxia or high environment is interesting. My lab, I didn't talk about it, but we've had a long-term project looking at hypoxic signaling and aging and lifespan. And in C. elegans, it's absolutely the case that if you can modulate oxygen levels and find an optimum that is different from environmental oxygen that extends lifespan. So there could be impinging on hypoxic signaling. It could be dietary, and that's, you know, what most people will point to for most of these places, right? That it's either something like a Mediterranean diet or a caloric restriction like diet. So the Okinawan islands off of Japan are a classic example where people think that that's a natural population of caloric restriction. And then getting back to the prior question, you know, there is also this stress-related component where there is evidence that chronic stress can accelerate some of these molecular biomarkers of aging. Epigenetic changes to telomeres. So some of these hallmarks of aging that we think a lot about can be impacted by chronic stress, and stress can come from a variety of places, but you know, socioeconomic status certainly has been associated with shorter lifespan and more age-related disease. And so it seems likely to me that low stress environments, you know, you may actually get a benefit from that, at least from a lifestyle perspective. So I think all of these things are potential contributors, and there's not a lot of great data to try to figure out what the actual mechanisms are at this point.
There's a question in the chat regarding, can we get a link to the ongoing dog study? This person, Mario, would love to share it with some friends.
Yes, so dogagingproject.org is the website. So that's probably the easiest way. But certainly shoot me an email if that's not answering the question. I'm not sure exactly what's being asked, but if that isn't enough information, feel free to send me an email. My email's right here. And I will put you in touch with the appropriate person if there's more information desired.
Dr. Kaeberlein, I have one more question. Some of the most intrepid and fearless scientists believe in their own science. And I'm fairly certain you have some rapamycin in your lab. So the question becomes, are you doing the experiment on yourself? And you don't have to answer that.
Yeah. I mean, you know, I get asked this question all the time, as you might imagine. And my answer is not currently. So the one thing I will say, because I get asked this question all the time, is I certainly, first of all, I do not recommend that people go out and take rapamycin. I don't view that as my job, right? I'm a research scientist. I don't have any problem with self-experimentation, but I'm not going to get in the habit of telling people what they should be doing until I feel like the data's there. So, and people ask me, you know, should I buy this rapamycin from this you know, company in India that's research grade and put it on my cereal? And no, don't do that. But I get asked that question all the time. So I would say personally, and I've already said this, I think that when taken at low doses, intermittently, rapamycin is extremely safe. And I would personally have no concerns about doing that myself. And I may or may not have done that in the past. And I'll leave it at that. But I don't recommend, I'm not recommending other people do that. And if you do it under the supervision of a physician.
However, Matt has not changed. I met him 21 years ago. I think
That's
he's
not
doing
quite.
something secret that don't reveal. Yeah, he did not change. He did not change. Something is
Fortunately,
secret.
we don't have the group picture from that. I think both of us.
Yeah, that's what I'm referring to. Yes.
I see a hand up.
Dr. Sarsour, go ahead.
Yeah, great presentation. Thank you for this presentation. And I agree with you. A lot of diseases are funded, but aging is not looked at as a disease. And to be honest, I work on aging too, but until I stuck cancer there in my research, I never got funded. I get funded by private foundation, but not really. So that's great. So my question is, did you see any metabolic changes? Did you examine for metabolic changes when using rapamycin? Because mTOR really affect every metabolic pathway. And I wonder which pathway you are seeing if you did that. So that's one thing, you know, I'm really wondering about.
Yeah. Yeah, it's a good question, and it's extremely complicated. So I mentioned the mitochondrial disease model. So that's a complex 1 deficient mouse. And there we absolutely see profound metabolic changes. It rescues the disease. I mean, doesn't cure it, but those mice live about 3 times longer than they did previously. And we think that's at least in part through fat mobilization and ketogenesis. So at high enough doses, rapamycin will promote fat mobilization in mice and ketogenesis. And we think that's part of the story there. We also work, this is unpublished data that hopefully will get published soon, in a diet-induced obesity model in mice. And again, at high enough doses, rapamycin prevents diet-induced obesity. We think it's promoting fat mobilization and fat metabolism. And we think it's mediated, at least in part, through a transcription factor called CEBP beta. So this is, there's a lot of work from Cornelis Kalkoven's lab on CEBP beta and fat metabolism. It's actually a really interesting transcription factor that's regulated through a translation of an upstream open reading frame by mTOR. So when mTOR is inhibited, you get more translation of the active form of CEBP beta, and that promotes fat metabolism. And so we think that's part of how rapamycin is affecting fat metabolism, at least in the context of diet-induced obesity. So I agree with you. I think that it would be shocking to me if some of the effects we see in the context of aging from rapamycin and mTOR inhibition are not mediated through these metabolic intermediates. But it does so much that it gets hard to really focus in on one thing and figure out what the details are. But I think CEBP beta is one place to look. For a specific downstream target.
Thank you so much, because when you said fat metabolism and lipid metabolism, that really make me so happy, because in my work I did some RNA sequencing, and one pathway really pans out in my research is lipid metabolism. When I compare different fibroblasts from different aging groups, it's always pops up is when I do the RNA sequencing and all, it was lipid metabolism, and that made me really happy now. and thank you for the information, because I'm going to use that in my grant.
Yeah, you absolutely should. I'll give you one more tidbit that I always thought was interesting. So there was a study that looked at 31 recombinant inbred lines of mice and subjected them all to caloric restriction. This is work that Jim Nelson and Tom Johnson and Brad Rickey did. Chen-Yu Liao, Liao, is the first author. So the question is, how does genetic variability affect lifespan extension from caloric restriction? That's how they started. And what they found was that about half the recombinant inbred lines had their lifespan shortened, some had no effect, and some had lifespan extended. The reason why this is relevant for this conversation is in a subsequent paper, when they asked, what was correlated with that effective lifespan from caloric restriction, the strongest predictor of lifespan extension from caloric restriction were those lines that maintained body fat under caloric restriction. So there's some key connection here between metabolic response to caloric restriction, which of course turns down mTOR in addition to other things, and ability to maintain body fat. And as far as I know, nobody has figured out what that connection is. So it's a really interesting unanswered question, which I think is going to turn out to be really important when we start to try to translate some of this stuff, because humans are genetically variable, right? These interventions are not going to work the same way in every person. And so we need to understand what are the mechanisms that lead to genotype specific responses to caloric restriction or to rapamycin. And I think this is a really important clue.
Wow, this is fantastic. And maintaining body fat, that's basically one of the players I'm working on is actually maintaining a balance between lipolysis and fat droplets. And this is fantastic. Thank you very much for the information, and then really great. Thanks.
Okay, Dr. Nicole Ford, I believe, will take over the next part of us hosting you here at KCU. Dr. Ford, are you in the room?
Yes, I'm here.
Take it away.
Stop sharing my screen now, unless there's any reason to do that. I can try to stop sharing it. There we go.
And I'll stop recording now.